Okay, yeah, go for it, John, on the start webinar. And then, like I said, just fade the music up more and more as you sort of up until 1 minute past, and then you can fade it away. Okay, yeah, go for it, John.
Good morning, all, and welcome to the second Grand Rounds lecture of 2026. These Grand Round lectures are sponsored by the Shanks Foundation and also the Pathological Society. I am the president of PathSoc, and it gives me great pleasure to introduce today's speaker, Dr. Matt Clarke, who is a clinician scientist and consultant pediatric neuropathologist at Greater Ormond Street Hospital and also based at the Institute of Cancer Research. He trained at Keele Medical School and completed a PhD in molecular pathology of infant gliomas. Matt is also former chair of the college's trainee advisory committee, and also he's involved in the Academy of Medical Royal College's trainee doctors group. He is currently the BDIAP meeting secretary and former editor of the ACP News. Matt is incredibly engaged and a very busy person. He's received a number of awards for all of his public engagement activities, for his research and science communication. And he featured also in the Pathology Power List 2021. So it's with great pleasure that I hand over the podium, so to say, the virtual podium, to Matt, who will be speaking today with the title of his talk being Beyond Histology, Revising the Classification of Infant and TYA high-grade gliomas through molecular profiling. Thank you very much, Matt. And before, just to ask everybody to please note that there is a Q&A button at the bottom of their screens. So please feel free to start asking questions throughout Matt's talk, and we'll take up as many as possible at the end. Thank you very much, Matt.
Thank you, Sarah. I'll just share my screen. So good morning, everybody, and thank you very much for that kind introduction, Sarah. It's a pleasure to be with you here this morning, and thank you very much to the Pathological Society for inviting me to be a part of this March Grand Rounds. And as Sarah said, I'm going to talk to you today about Beyond Histology, Revising the Classification of Infant and TYA high-grade gliomas through molecular profiling. So just to run through what I'm going to talk about throughout this talk, I'm going to talk about my work that formed the basis of my PhD project about infant high-grade gliomas. And for those who may not be so familiar with pediatric high-grade gliomas, I'll give you a little bit of background to this work and then talk to you a little about the different methods that we use to help classify and characterize these tumors and the significant clinical impact and future research questions that have come from this work. Moving on from that, I'll talk to you a little bit about the work I've done post-PhD, working on another group of understudied tumours in high-grade gliomas in teenagers and young adults, and talking about some of the TYA-specific high-grade glioma subgroups that have come from this work, and again, the opportunities for targeted treatments that are available now. And then looking to the future, I'm looking at some new potential high-grade glioma subgroups that I'm working on at the moment through my clinical work, talking a little bit about post-mortem sampling related to these tumours, and also some of the new techniques that are supporting diagnostic pathways and research going forward as well, which you might be interested to hear about. So I appreciate that I'm not talking to a neuropathology audience. So to give you a little bit of background around the issue of high-grade gliomas, they represent a significant clinical problem, and they can occur in patients of any age, right the way down from infants all the way up to the elderly populations. And they can occur in a range of different anatomical locations, including the cerebral hemispheres, the brainstem, the cerebellum, and the spinal cord. And we know that tumours that are located in the midline or the brainstem tend to occur in patients of a younger age. But irrespective of their location, the prognosis is dismal for these patients, with around a 2-year survival after diagnosis. So it really represents a significant clinical challenge. And this is particularly so for those tumours located in the midline and the brainstem, because at the moment, the gold standard treatment for these tumours is surgery, and therefore those tumours located in the brainstem and midline are not amenable to surgical resection because of the risk associated with potential damage to the important structures which we need to survive in those regions. And so we're reliant, therefore, on things like chemotherapy, which in very young children has cumulative damaging effects on the developing brain, and also the challenges of chemotherapy, where we have to overcome the blood-brain barrier as well. So it really is a significant clinical problem, which really is in desperate need of continued research to support as well. But there is also the biological side of these tumours, and they're not just one entity, they represent a huge diversity of different subgroups with different distinct genetic and epigenetic driving alterations. And this is the visual abstract from our paper of meta-analysis that was published in 2017, looking at over 1,000 different examples of high-grade gliomas, attempting to group them according to the different mutations they contain. And we see a portion of them that are characterized by distinct histone mutations, K27M or the G34 alterations, others that have IDH1 mutations, others that have a mixture of MAPK pathway alterations. But also you'll notice down the bottom here, this bar described as wild type or grey. And again, this represents tumours that we still don't know a lot about. And each year, thanks to the increased work of molecular profiling and research, this bar is getting narrower and narrower. And we're currently undertaking a repeat meta-analysis of over 2,000 cases this time, looking at now the changing the spectrum from the time of this paper being published. But it's not also the type of mutation that they have, it's also the number. So we have tumors within the subgroups that just have a single driving alteration, and we have others that represent some of the highest mutational burden in human cancer, with thousands of different mutations in the hypermutator sort of phenotype. And so you can see that this represents a significant biological problem in how to actually deal with these tumors and treat them effectively. Now, high-grade gliomas in children and young adults are distinct from those occurring in the adult population, and part of the distinction is between their mutational profile, with around 50% of pediatric cases at least containing some form of histone mutation. And there are two main groups that are now recognized by the WHO classification and defined by these different histone alterations. So the K27M altered tumors are where we see mutations in the H3F3A or HIST1H3B genes coding for the H3.3 or H3.1 histone. And we see a change at position 27 where a lysine is converted to a methionine. And interestingly, these tumors are defined by a midline location, so presence in midline structures such as the thalamus, the spinal cord, or other brainstem structures. Whereas the G34 mutant tumors, where we see at position 34 a change from glycine to arginine or valine in the H3F3A gene, these tumors are characterized by a location within the cerebral hemispheres. And we know that the K27M mutant tumors tend to occur in patients of a younger age group, but unfortunately, irrespective of the histone mutation these patients have, the prognosis is dismal, even compared to the wild type tumors. But as I mentioned, in the 2021 classification, a step forward has been taken now in that these tumors are recognized and defined by the molecular alterations that they contain. But what about infant gliomas? Well, these are occurring in patients under the age of 1, with around approximately 2 to 3 new cases occurring in the UK a year and around 15 across Europe. So they're quite rare, but they again can occur in any location within the central nervous system. But what has been interesting is that for some years, they've been known to have a slightly improved outcome compared to other high-grade gliomas in pediatric cases. And there have been odd case reports that have been published showing increased survival associated with these, which is different to what we know about with low-grade gliomas, which actually have a worse prognosis in the infant population. So there's been a drive for some time.
Sorry, we seem to have
Jones,
lost.
I undertook a project and then progressed into my PhD, which aimed to explore this particular question. And so I gathered, thanks to one under the age of 4, CNS WHO grades 2 to 4, encompassing glial and astrocytic and glioneuronal tumors, but excluding other well-characterized entities such as pilocytic astrocytomas, ependymomas, and embryonal type tumors. And then through a series of different molecular analyses, we were able to try and refine this cohort down to the group that we were particularly interested in. And one of the key bits of work that we did was using methylation profiling in order to try and exclude some of the other well-recognized high-grade glioma and CNS tumor entities, and bringing this down into a cohort of around 130 cases, which we termed the intrinsic set, which didn't really fit with any of the other known entities at all. And then we could use other techniques like fusion panel sequencing and whole exome sequencing, copy number profiling, as well as a pathological review of these cases to understand exactly what these tumors actually consisted of, both histologically and molecularly. For those who are not so familiar with the process of DNA methylation profiling, I just wanted to give you a little bit of background to this work. And this all stemmed from a seminal paper by David Kappa that was published in 2018. All of our DNA is methylated to some degree, and these little methyl groups are positioned at sites known as CPG islands. And what the paper published in 2018 identified was that if you take the DNA from all the known types of brain tumors at that time and look at the profile of where these methyl groups positioned, each brain tumor had a different profile of methylation, which represented a tantalizing opportunity of using this technique as a tool to help guide neuropathologists. And so this led to the development of the Heidelberg brain tumor classifier, where you can actually sequence it by an array-based sequencing technique, looking at analyzing where these methyl groups are added, put your data through the classifier, and then by comparison with a reference cohort of excellent examples of the known brain tumors, it will give you an output of a calibrated score from 0 to 1. 1 being it fits with this particular type of tumor, and closer to 0 meaning it doesn't really fit with anything within the classifier. Those we usually take diagnostically a score of 0.87 to be diagnostic, but even those cases that have a lower score still can be reliable or useful in terms of diagnosis, because there are various factors that could influence that score, including the cellularity of the tumor, but also the quality of the DNA as well. And the image on the right-hand side here, this is a TS&E plot. And this is a way we can actually visualize methylation data. So each color represents a particular tumor type. So the ones in yellow that I'm circling here are IDH mutant tumors, and each dot represents an individual case. So you can see that these cases in the reference cohort cluster together by their methylation profile. And so we can add our cases in here to see which one they land with, and then compare that with the calibrated score that we get from the classifier. But you will also notice there's an awful lot of white space around here as well. And sometimes when adding cases into this, you find that cases will land in that white space, and over time, more and more may cluster within that location. And it can also therefore be useful as a research tool to discover new potential subgroups of tumors, but also you can start to see patterns emerging within an existing entity itself. And so what was originally termed one single group may in fact be made up of lots of different subgroups. So it is a really valuable tool, both in terms of diagnosis and research as well. Another important thing that the output from this study was actually improving the accuracy of diagnosis for these tumors, and around 12% of cases in this cohort actually had some form of change in their diagnosis. So some tumors actually went from low grade up to high grade, and others went from low grade down to high grade down to low grade. And this is really important because there are huge treatment implications for these different graded tumors. And so particularly in pediatric cases, we want to make sure that we're not over treating cases and leaving these children with some sort of cumulative problems and later on in life as well. But also as neuropathologists, in our desire to provide as accurate a diagnosis as possible, this technique can be very useful to help refine that. Now, as I mentioned, when I gathered this cohort of 241 cases, and through methylation profiling and exclusion of other well-characterized entities using our CNS reference cohort, we were able to refine this down to 130 cases that didn't really fit with any of the other entities. And when I plotted this into this TS&E plot of a reference cohort of just glial, high-grade glial and low-grade glial tumors, you can see that they clustered together in this separate group away from the other entities, but nearby to another well-known type of infant-type glioma called the desmoplastic infantile ganglioglioma and astrocytoma. But the most majority of these were a new type of tumor, which was now called the infant-type hemispheric glioma, which was, at the time, nothing was known about these tumors other than the fact that they seemed to be showing this distinct methylation profile from the output of this classifier. So my next steps were then to try and work out what these tumors actually consisted of. Looking at the epidemiology of them, they again started to show some interesting patterns. So in terms of their location within the brain, if you look where the dark brown circles are, the vast majority of them were located in the hemispheres, either the frontal or the parietal lobes most frequently. There was a slight male predominance associated with the infant-type hemispheric gliomas, but what was also very striking is that these seemed to be a tumor that occurred in patients under the age of 1, with just a handful of cases occurring in children older than this, the highest being a child aged 2.5 years. Copy number data is also something that we can get from methylation profiling, so a sort of 2-for-1 from the test. And what we usually expect to see with high-grade gliomas is a lot of different copy number changes across the different chromosomes. And this is a heat map of the data for the intrinsic set of cases. And you can see, for those who haven't seen one of these before, this is the chromosomes labeled down the bottom here, along the side here, and each column here represents an individual case. And what you can see, which is interesting, is that around 2/3 of this intrinsic cohort showed a silent copy number profile, which was quite unexpected, with around 1/3 of them showing some subtle alterations, but not anything particularly common across this intrinsic cohort at all. In terms of the histology of them, again, this was something as a neuropathologist I was particularly interested to look at. There were some subtle features defining this infant type hemispheric glioma. So one of them was the increased cellularity and the architectural sheet-like appearance. So there was very little variation in terms of their architectural layout, just a very highly cellular tumor. In terms of the cytology show, there was some variation. So some of the tumors show a spindle cell appearance where the tumor nuclei are very elongated, and others had a more gametocytic appearance where the tumor cells had plentiful eosinophilic cytoplasm with a more rounded nucleus. And others rarely had focal areas of ganglion cell differentiation. And I'll talk to you about the significance of that later on in the talk. But also there was no doubt that they had the typical high-grade histological features of a high-grade glioma. So the presence of necrosis, the presence of microvascular proliferation, and also frequent mitotic activity as well. And when looking back at what tumors were reported as originally at diagnosis, 84% of them were reported as some form of high-grade glioma. So again, predicted to have a very poor outcome. But when we looked at the survival data for these, what was really striking is that they showed an significantly improved survival compared to other high-grade gliomas occurring in this age group, following a trend more in line with the CNS WHO grade 1 tumors. So this really brought into question, here we have a histological tumor that looks high grade, but clinically is behaving much better than predicted. And so therefore we're bringing into question the CNS WHO grading system, particularly for this particular tumor. So then I wanted to look a little bit more about what the molecular characteristics of this tumor was. And so then we turned to sequencing, so using panel and whole exome sequencing to look at some of the genes that we typically see mutated associated with high-grade gliomas in pediatrics. And if we look at these other glioma subtypes which were occurring in the infant population, we see the usual suspects appearing. So alterations in PIK3CA, some histone mutations, and PDGFRA as well. But when looking at the intrinsic set, with the majority of which were the infant type hemispheric gliomas, it's a very different pattern. So just a couple of NF1 alterations, a TP53 and a PIK3CA alteration. So nothing in common, just very quiet sort of profile here of somatic alterations. But when we did some fusion panel sequencing, the pattern was very different. So these tumors were characterized by a form of RTK fusion, either in ALK, ROS1, MET, or NTRK. And this seemed to be the only alteration present within these tumors, and therefore was driving this particular tumor. We also saw a few fusions present within the other glioma subtypes, but interestingly, these didn't appear to converge a survival advantage on them, and they often didn't occur in isolation and occurred with other types of mutations as well. So around 80% of these intrinsic infant type hemispheric gliomas contained one of these fusions, but there was interestingly also a portion of these which were fusion negative as well. The NTRK and ALK were the most frequent fusions that we identified, and there were lots of different fusion partners that were identified with these as well. And although limited in terms of number, there did seem to be some differences in survival associated with the type of fusions. So the ROS1 fusion seemed to be doing better. And interestingly, the fusion negative cases, which we did some whole genome sequencing on and still were not able to identify a fusion present within them, seemed to have a worse outcome overall. It still remains my hypothesis that these cases still have a fusion within them that's driving them, but we weren't able to identify what that was in the duration of this particular study. But it's interesting to see that there are possible differences in terms of these different subgroups. The methodologies that we used to identify the fusions were variable. So we had a custom-made DNA fusion panel, which the results of which we validated with Sanger sequencing, but we also were able to screen cases by looking for changes in copy number. So for example, ALK fusions often were suggested by a little bump associated with chromosome 2 on an otherwise flat copy number profile. And we also saw changes associated with the ETV6 NTRK3s as well. So again, representing a possible screening tool that you could use for analyzing your copy number data before then proceeding on to do RNA sequencing, etc., to validate the presence of a fusion as well. There also seem to be some interesting differences in terms of the proliferation index associated with the fusion positive cases. So when doing a Ki67 proliferation index, it was noted that there was a significant increase in the Ki67 proliferation in the presence of particularly NTRK3 fusions compared to the fusion negative cases. And there was some suggestion of this in the ALK fusion cases as well. So again, it seemed to be these were more proliferative in the case of a presence of a fusion. We also went on to look at some epigenetic alterations in fusion positive and fusion negative cases, and using methylation-based gene ontology analysis of differentially methylated regions, there was no particular overlap between those in the ALK, NTRK, or fusion negative cases. But what we did identify, particularly with the ALK, was the dysregulation of genes associated with morphogenesis and development, glutamate receptor activity, and also synapse assembly. And for the NTRK fusions, dysregulation of genes associated with the J and K cascade, embryogenesis, and neuronal differentiation. And for the fusion negative cases, stem cell fate, TGF beta signaling, and pattern specification as well. We also noticed that there was a reduction in the methylation of CpG sites that were governing the expression of Wnt5a in the ALK fusion cases and STAT1 in the NTRK fuse cases. And we were able to show differential expression, protein expression of these two using immunofluorescence. And then finally, we used the nanostring assay for looking at 30 of the most differentially methylated genes, and we were able to distinguish show differences between the fusion positive cases in the specifically the ALK and NTRK and those that were fusion negative as well. So there were differential epigenetic regulation of key genes and protein expression, particularly characterizing the NTRK positive, ALK fusion positive, and the fusion negative cases. We also wanted to then go on and look at doing some in vitro and in vivo studies, looking at the significance of these fusions that we'd identified. And thanks to national and international collaborators, I was able to establish 2 patient-derived cell cultures that were sufficiently established that we could enable us to do different assays associated with them, both of which contained NTRK fusions. And when testing them in vitro with different TRK inhibitors, so entrectinib, crizotinib, and milsiclib, they showed sensitivity to these inhibitors when compared to fusion wild-type cases of other examples of pediatric high-grade gliomas. And thanks to our collaborators with a team in Heidelberg, we were able to use in utero electroporation techniques to develop a in vivo model of the most frequent ALK fusion we identified, the PP1CB ALK. We're able to develop this model and also show that sensitivity to the treatment with an ALK inhibitor, lorlatinib, and again, so validating the importance of this fusion within these cases, but also showing how important it is in terms of potential clinical translation as well. So in relation to that clinical translation work, what was such a privilege of being part of this study is that we were able to influence some of the real-time treatments of children that have been diagnosed with these particular tumours. And these are 2 such examples from the paper that we published. The one on the right-hand side was a little baby who was diagnosed before she'd even been born with a tumour. And a few days after she was born, she underwent a biopsy that confirmed that this was a high-grade glioma, so predicted to have a very poor outcome. A short time after that, she was treated with chemotherapy and then underwent further surgery. And thanks to our collaboration with the team in Italy, a small sample of this tumour was sent to me at the lab, where I was able to perform these analyses and confirm methylation was an infant type hemispheric glioma and also contained an ETV6 NTRK3 fusion. Despite the best efforts of the surgeons, they were not able to resect all of the tumour indicated by the red outline in this on this scan. And thanks to this, identifying the fusion, she was initially started on crizotinib, which was followed up with larotrectinib. And as you can see, subsequent scans showed the shrinking of this tumour. And I'm delighted to say that she is alive and well and disease-free today now, and 7 years old and at school and doing remarkably well. And there are other cases now of these children who are surviving and where this disease is being cured as a result of this work as well. One of the other things that we were able to collect as part of this cohort were a small subset of longitudinal cases where there have been several resections that have been done of these tumours as well, which also added an interesting element to my PhD project. And you can see from these different swimmer plots the variety of different treatments that have been used, often chemotherapy, and in line to try and treat these tumours. But in some cases as well, going back into the archive, it was very interesting to see that some young babies who had a biopsy and were confirmed to have a high-grade glioma, unfortunately, because of the poor prognosis associated with these tumours, often at that point, they would be referred to palliative care because the prognosis was so dismal with them. So it's really important that we can now showcase that something can actually be done to treat these tumours going forward. In terms of looking at comparing the initial biopsies and the longitudinal samples, we found that the methylation profiling was consistent between the two samples. So there was no difference in methylation. But we also did identify a few different variants that were picked up across this longitudinal cohort that weren't present within in the initial sample. But again, the caveat being that we didn't have the germline to actually compare to with this. But the key thing, that an important message that was that none of these genes have been particularly associated or reference what these are doing. Is it remains in copy number. So some showed similarities between the first and the second resections, but also a few had subtle changes associated with the copy number as well. So there were some differences in the somatic mutations and copy number, but the methylation was maintained across the cohort. But what I found really interesting from these tumours is the histology of them. So this was one such example of a typical appearing infant-type hemispheric glioma with a very cellular tumour, pleomorphic-looking nuclei, some spindling to them, and sheet-like architecture with them. But when looking at the longitudinal sample, there were differences in the morphology. So some areas had more spindle-like nuclei, but also other areas had quite a lot of ganglion cell-like differentiation. And I showed you one in the initial histology panel that had a similar appearance to this. And so what I think is happening here is this is evidence of potential histological differentiation of these cells in some of the longitudinal cases. And also we have some clinical and in vitro anecdotes associated with this. So the patient-derived cell cultures that I was growing would often reach to around passage 10, and then the cells would start to become bigger and would slow down in terms of their divisions. And so by the 11th or 12th, we wouldn't be able to expand the patient-derived culture any further. Also, in the clinical setting, there were several examples where patients had undergone a resection that not all of the tumour could be resected, but they hadn't been treated with anything else and were just being watched and waited to see what happened. And these tumours have remained static as they were and haven't actually progressed. And for a high-grade tumour, that would be extremely unusual. So again, I wonder whether what we're seeing in these histological images here is what we may be seeing in the cell cultures, but also clinically as well. So the culmination of this work led to the development of this classification of infant high-grade gliomas, recognizing that there are some of the well-established types of high-grade glioma that occur within the infant population, but still have a very dismal prognosis. We have the desmoplastic infantile ganglioglioma and astrocytoma are an already well-characterized entity that characterized by MAPK pathway alterations and known to be associated as an infant tumor. But we now have these other tumors that form the basis of the intrinsic set, the infant-type hemispheric gliomas, that are characterized by some form of RTK fusion, but also this intriguing set of fusion negative cases where we weren't able to identify a fusion that was driving these or any other mutation associated with this, which again represents a really interesting opportunity for further study as well. From this work, the 2021 classification, WHO classification, now included a chapter on the infant-type hemispheric glioma to ensure that these are now recognised nationally and internationally, and guidance associated with how to diagnose these. It also led to the CONNECT consortium to develop a clinical trial, which is known as the CONNECT 1903 clinical trial for such patients that are identified to have an RTK fusion diagnosed with a high-grade glioma. And also from this, we started to look at other groups of understudied high-grade gliomas, and particularly focusing on teenagers and young adults. And with the support of Rita Pereira, who was a scientific officer in our team and now is a PhD student, we were able to take this further. And I'll talk to you a little bit about that in the next part of the talk. But I also wanted to talk to you and mention another intriguing question that has come from this and been highlighted very recently. And if we go back to our original TS&E plot, there was a very close relationship, as I mentioned, or certainly visual relationship between the infant-type hemispheric gliomas and the desmoplastic infantile ganglion gliomas, with a spectrum of tumours that seemed to form a trajectory between the two. And I often wondered whether, in fact, there was a relationship between these two entities. And in my clinical practice at Great Ormond Street, we've come across, myself and colleagues have come across a couple of cases recently which seem to possibly suggest this. So these were two tumours in patients under the age of 1 who presented with seizures with large hemispheric tumours that were subsequently resected. Histologically, these tumours showed features typical of a glial or glioneuronal tumour in the spectrum of those associated with a desmoplastic infantile astrocytoma or ganglioglioma. But interestingly, they had focal areas where the atypia was more marked, and the mitotic counts and the Ki-67 proliferation index was also very high, which was an intriguing feature of a tumour that is considered a CNS WHO grade 1 tumour. And when looking at the molecular profiling of these tumours, interestingly, both of them classified as infant-type hemispheric gliomas by methylation profiling with, as you can see, a very high score, and also showing the typical flat copy number profile that we would expect with these tumours. But what was also very striking is that they both contained a RAF1 fusion, which are known to occur within the desmoplastic infantile astrocytomas, a MAPK pathway alteration that would be typically seen with them. So here we have a tumour that histologically and with the identification of the fusion looks more typical of a desmoplastic infantile astrocytoma, whereas the methylation profile and the copy number profile and potentially these higher grade features on the histology suggest an infant-type hemispheric glioma. So further work is ongoing now with collaborators in the Netherlands to try and refine and understand a bit more about this cohort with some other different molecular techniques as well. So just to summarize the infant work that I talked about, these infant gliomas comprise multiple different biological and clinical pathological subtypes with few copy number alterations, somatic mutations. They're occurring in children under the age of 1, and they're characterized by some form of RTK fusion. And they also have these characteristic histological, architectural, and cytological appearances. They show improved survival compared to other high-grade glioma subgroups under the age of 4. And the in vitro and in vivo data suggest sensitivity to TRK inhibitors, which has then led to the development of the CONNECT 1903 trial as well. They were included as a new high-grade glioma subtype within the WHO classification 5th edition, the infant-type hemispheric glioma. And I'm delighted to say that I'm now the responsible author for this chapter and the desmoplastic infantile astrocytoma chapter in the forthcoming 6th edition of the classification, which is due out later this year. And this future research question is very intriguing about the possible relationship between IHGs and the DIA-DIG, and really important to answer because clinically, one is diagnosed as a CNS WHO grade 1 tumour, and the other is considered still part of the high-grade glioma spectrum. So for clinicians, this is really difficult in how to actually treat some of these tumours which have overlapping features. So I think further research in this area can really help. Moving on to high-grade gliomas in teenagers and young adults, you can see here from the 5th edition classification that significant steps forward have been made in terms of the definition of tumours because they are characterised by their molecular alterations actually by definition. And you'll also notice that we have adult-type gliomas and also the paediatric type, but it's still not recognised what about the teenagers and young adults and where do they fit in the spectrum of these different tumours. And the biggest study at the time that looked at a cohort study of these tumours and identified that we see the usual suspects occurring in this population. So some tumours with IDH mutations, others with histone, and others with MAPK pathway alterations. But around 50% of them were still considered not elsewhere classified or not otherwise specified. And so they really represent an understudied group of tumours that I wanted to focus a bit more on and understand more about. So in a similar vein to what I'd done with the infant project, I gathered 207 cases from national and international collaborators from patients aged between 13 and 30 from any CNS locations across CNS WHO grades 2 to 4, but importantly excluding the well-characterized high-grade glioma subtypes, so making sure they were IDH wild type and histone wild type. And then using methylation profiling again and comparing with our CNS reference cohort and our glioma reference cohort, funneling these down into a core set of cases with novel, potentially novel subgroups that we wanted to explore further with different sequencing such as whole genome, whole exome, fusion panel, and DNA copy number, and then a pathological assessment of these in parallel as well. And this is a pie chart showing the spectrum of different tumor subtypes that we identified as part of methylation profiling. And you can see that there are an awful lot more in terms of this, a variety of different subgroups as part of this IDH wild type and histone wild type category. And there are also still a proportion of cases that didn't classify as anything by DNA methylation profiling, but are not smaller compared to what was originally published in the original cohort study. And when plotting these into our CNS reference cohort of over 8,000 cases, you can see that they cluster within the central area representing where the high-grade gliomas are more frequently seen. And looking at this in terms of our glioma reference cohort, we again start to see some interesting features. So the circles are highlighting cases that are classifying as some of the adult type high-grade gliomas, including the GBM-RTK1 and the GBM mesenchymal subtypes. Interestingly, only 3 cases went into the cohort that were diagnosed at the time as anaplastic pleomorphic xanthorastrocytomas, but around 25 cases actually classified as this particular subgroup. Also, around 2/3 of cases classified as some form of pediatric high-grade glioma, including some novel subgroups known as the HGGB, HGGE, and GBM-CBM group, where at the time next to nothing was known about these other than the fact that they had a distinct profile by methylation. What we also did was gather all the publicly available methylation data that was available across high-grade gliomas and plotted them into this age density plot. And you can see here the boundary set out for 13 to 30, which was the cohort that I was looking at. And it suggests that particularly this novel subgroup, the HGGB, seemed to be a TYA specific type of high-grade glioma, with other cases, other subtypes having enrichment within this particular region as well. But what I also thought was very striking is that when we look at the pediatric subtypes, we see the expected peak in the pediatric location, but also we see peaks occurring in the adult population as well. And I think at the moment, certainly standard of care is for all pediatric cases to have DNA methylation profiling. But I think this is a further argument to keep striving that all patients for all brain tumors will have this test done, so that we don't miss these interesting pediatric type tumors that are occurring in older adults. And this will be important for future research, comparing the 2 tumors in these different populations and seeing what may be different about them. Looking at the copy number data for these different subgroups, we again started to see some patterns. So for the MYC-N amplified cases, there were alterations associated with chromosomes 1 and 2. And for the GBM mesenchymal groups, there were gains of chromosome 7 and losses of chromosome 10. Amplifications most frequently consisted of the PGFRA and EGFR, and CDKN2AB deletions were the most frequent, which wasn't a surprise given some of the subgroups that we had within this. In terms of the whole exome data, there were characteristic somatic variants that were frequently associated with high-grade gliomas, including the TP53, ATRX, and PDGFRA, to name a few. And we also did some fusion panel sequencing, this time using the ArcherDX fusion panel. And other than a single case of a ROS1 and MET fusion and a BRAF fusion, which characterizes a particular tumor known as the high-grade astrocytoma with pyloid features, recurrent fusions were not associated with TYA-HGG, different to what we had already seen with the infant population. When looking at the sequencing data in terms of the methylation classes, we again started to see some interesting findings. So for the adult type tumours, these were enriched for RB1 and PTEN alterations. For the well-characterized groups such as the PXAs and the high-grade astrocytomas with pyloid features, these were characterized by CDK and 2AB deletions and MAPK pathway alterations, most frequently the BRAF V600E, which was already well known at the time. The pediatric high-grade glioma subgroups were enriched for T53, ATRX, and PDGFRA, and the MICN, unsurprisingly, had associated with MICN amplifications. But an interesting finding that came out from this was that there were a cohort of cases that had a hypermutator phenotype, enriched for the novel HGG-E subtype, but also identified across other different subtypes as well. And this was really important to identify because these patients could be amenable to treatment with checkpoint inhibition. So to show that they occur within the TYA population was an important finding. Looking specifically at the HGGB subgroup, which was suggested from our methylation data and the age data that it was a TYA specific subtype, there was no particular gender predominance with them, and they showed a variety of different locations within the central nervous system, most frequently, though, hemispheric location in the parietal lobe. But in terms of the histology, these are 6 cases that scored highly as the HGGB subgroup by methylation profiling. And you can see the variety of different histological appearances they had. Some showing this spongiotic type appearance, others showing a more oligodendroglial-like appearance with a rounded nucleus and clear cytoplasm, others with a more fibrillary stroma, others with more desmoplastic stroma, and some showing a more infiltrative glial phenotype. The important message here is that phenotypically, just by looking at this, we wouldn't be able to reliably diagnose this as an HGGB subgroup, and showing how important it is that molecular profiling has done all these cases to understand the diagnosis properly and provide an accurate diagnosis in these cases. Sadly, although limited in terms of number, the survival of these cases was very poor. There were no particular copy number changes that they showed in common, but what they did show was an enrichment for MAPK pathway alterations and also amplifications in genes such as EGFR, MET, and CDK4 as well. And shortly after we published this paper at the beginning of last year, another group in the States also published a study focused specifically on this this particular methylation subgroup, calling it the diffuse astrocytoma adolescent and young adult subtype with MAPK pathway alterations, otherwise known as DAYA, validating the findings that we had already published related to this study as well, and will certainly be under consideration as a potential new subgroup for the WHO classification going forward. The other subgroups that showed enrichment within the TYA population were the GBM-CBM group, so named because of a predominance location within the cerebellum, showed a more oligo-like appearance histologically. And the GBM mesenchymal group again showed typical high-grade, marked high-grade glioma features, but also focally showed some areas of lymphocytic infiltration in places as well. But we also looked at differences in copy number by age because these tumours were not specific to the TYA population. So we compared those that were aged under 30 with those that were aged over 30. There weren't particularly patterns associated with the GBM-CBM group, but when we looked at those aged over 30 for the GBM mesenchymal atypical group, we showed a portion of these had gains of chromosome 7 and losses of 10, a typical profile that we would see associated with glioblastoma IDH wild type cases. But those under the age of 30 showed gains of chromosome 7, some alterations in chromosome 1 and also in 6, but not associated with chromosome 10. So there were differences in terms of the copy number profile and the age of the patient at diagnosis as well. We were also able to identify that there were some tumor syndromes that underlie the development of TYA HGG, with this showing a couple of cases of patients with CMMRD, both actually siblings that both classified as different types of high-grade glioma by methylation profiling, and also showing a hypermutator phenotype. But also, very sadly, we were able to identify that from the clinical histories, in 5 cases, patients have been treated in their pediatric years for some form of tumor, most frequently either ALL or a medulloblastoma, and then had subsequently developed a high-grade glioma in their early 20s and died as a result of that. And so we also need to be very conscious that in treating one cancer, we're potentially increasing the risk of developing a second high-grade tumour. And I advocated in the paper potentially considering the use of screening of these patients at an earlier stage to detect this deadly disease at a more early stage, where potentially an intervention may have a better outcome for these patients, but certainly something to increase the strive for kinder treatments that are not going to put a patient at risk of developing other tumours as well. So to summarize the TYA HGG work, they overlap with methylation subgroups which occur in both the pediatric and adult age groups, but there are several novel groups that show their peaks of distribution within the TYA age group. And the HGGB subgroup seems to be a TYA specific subgroup of HGG. They also show distinct patterns of copy number and somatic variants that characterize the different HGG subgroups, some of which may be susceptible to checkpoint inhibition, particularly the hypermutative phenotype. And I would also advocate that DNA methylation profiling, whole exome and panel sequencing should be performed on each high-grade glioma in the TYA age group to identify and characterize these potential novel classes, but also to identify those that may be susceptible to some of these treatments. For the last part of the talk, before I move on to a new potential subgroup, I just wanted to introduce this type of tumor, which is called a subependymal giant cell astrocytoma. It's a periventricular tumor that's composed of very large ganglion-like cells with these very pleomorphic nuclei, but strongly associated with a tumor syndrome known as tuberous sclerosis, but considered CNS WHO grade 1. And the reason for mentioning is that a paper in 2019 looked at 3 cases in the adult population that seemed to were periventricular glioblastomas that had an epithelioid or Sager-like morphology, but characterized by alterations in the MAPK pathway, TSC, and mTOR pathways as well. And again, in our clinical practice at Great Ormond Street, we've come across 3 cases, all aged between 3 and 12, that typically show the high-grade glioma features on imaging. But when looking at them histologically, again, typical architectural and cytological features of a high-grade glioma. But looking at the cytological features, they show interspersed cells that have typical appearances of a Sager, with very large, plentiful cytoplasm that's often glassy and very pleomorphic nuclei. Some scattered throughout the tumor, others having more groups of cells that having the similar appearance. And when doing the methylation profiling, they all classified as glioblastoma IDH wild type to varying different degrees, and particularly the atypical mesenchymal subtype, but not with particularly reliable scores. And again, it's very unusual to diagnose a glioblastoma IDH wild type in a pediatric case like this. And so I think these are falling into this subgroup because there's nothing currently within the classifier that better represents them. In terms of the copy number profile, they again showed some similarities. So all showed gains of chromosome 7, but also some gains of the higher chromosomes as well. And one case had a CDK and 2AB alteration. But when looking at the sequencing, they all showed variants in TSC1 or TSC2 alongside NF1 and TP53 alterations. So again, the TSC1 and TSC2 are usually features that we would see with Sagers. So the hypothesis is that these odd-looking cells scattered within these tumors may be those that contain these particular variants. So we're starting to see a somatic mutational profile of these tumors becoming apparent. But I also wanted to draw your attention back to our age density plot. And this is the group of tumors where these 3 cases classified with. And for an adult type tumor, what is very interesting is that there is a peak of distribution occurring in the pediatric population. And so what this potentially is highlighting is that this peak may represent other novel subgroups of these tumors of different types, one of which may be this Sager-like morphology that still need further work. And that's something that the team and I are going to be working on in the future to elucidate as well. So this new subtype seems to be characterized as TP53, NF1, and TSC1 or 2 variants, alongside this epithelioid Sager-like morphology, but also highlighting it's not just an adult type tumor, it can also occur in the pediatric setting as well. And the reason for the importance of looking into this further, these may be potentially amenable to mTOR inhibitors such as everolimus, and it may not be a cure, but it may provide some extra time, which of course would be extremely important in such a tumor with a poor prognosis. And using techniques such as single-cell RNA sequencing and spatial transcriptomics, which we're planning, may again help to confirm the relationship between these mutations and the phenotypic morphology of some of these cells as well. And just in the last minute, I just wanted to introduce some other work that I'm currently working on. And highlighted, I guess, by the PEACE study for the other cellular pathology specialties, there's been an increasing drive from parents and families wanting to donate brains to research. And this represents such a crucial window for us because at the time we usually just see the biopsy sample, possibly a longitudinal sample, then we don't know how the tumour has evolved at the time of death. And so there have been several cases over the years that have now been donated to us. And we're seeing increasing numbers more recently where we actually have the potential to do studies focusing on these multi-region areas, but looking at differences in terms of the histology, the standard of care testing, but also using other techniques like single-cell RNA sequencing and spatial transcriptomics on both non-tumor and tumor areas to look at different mutational spectrums, but also different morphological appearances as well, which is going to be really valuable in helping to understand more about how these diseases evolve. And certainly the next big thing in neuropathology that everybody is working towards at the moment, and certainly we're trialing at Great Ormond Street, is a new molecular technique, nanopore sequencing or long-read sequencing. One of the big challenges we have is that the turnaround time for our molecular work takes around 2 to 3 weeks. This could potentially provide a molecular diagnosis in a matter of hours and also intraoperatively as well. But there will be a huge volume of data that comes from this, including whole genome sequencing data that can be supplemented into research. And so I think this represents a really exciting opportunity to further enhance research into these particular devastating tumours as well. So in summary, significant progress has been made in the molecular characterization of high-grade gliomas. Molecular tests are an integral and essential part of the diagnostic pathway, both to bring together the integrated pathology diagnosis, but also expanding available tests and technologies that may help us to understand these tumours further. We're identifying new entities that are characterized by specific molecular alterations, which is providing future workflows for future research as well, but also represents significant challenges because although we're refining these cohorts down in the wild type cases into other smaller subgroups, it's still a real challenge for the clinicians in how to actually treat these. And so that's again a driver for further work. And also improving the speed of molecular results is going to be really important. And the advent of nanopore sequencing is going to help with this. But I also think we need to focus on using digital and integrating with AI to help, again, refine the accuracy of our diagnosis in the contents of these advances in molecular profiling. But importantly, again, relevant for all pathology specialties, improving data access, both clinical and molecular, for research is going to be a real priority. And there have been a lot of challenges related to this recently, but I think we need to work together as a community to help drive this forward to ensure that this can be relied upon and support research going forwards as well. So I'd like to thank both our national and international collaborators for their support of these various different projects, the funders, including the generous funding that I've received from the Pathological Society and the Shanks Foundation that have helped support this work. And also to the glioma team, in particular, Alan Mackay, Yura Grabowska, and Rita Pereira and Diana Carvalho, who have been instrumental in helping to progress this work forward, and to my supervisor and mentor, Chris Jones, who continues to be an active influence and helps to support me in all the work that I do in research and in my career as well. And just finally, just for those who are interested, I'm actually not at work at the moment and I'm not at home. I'm actually on leave and on a 5-day falconry course in Huntington. And these are just some pictures that I took over the last couple of days. On the left-hand side, you've got a great horned owl called Rimmel, who I was able to take out and walk around the park. And on the right-hand side, this is Pen, who's a red-tailed buzzard, who I've been learning to fly over the last few days as well, just in case anybody is interested. Thank you very much for listening.
That's brilliant, Matt. Thank you very much for an amazing talk and also for sharing with us your hobbies and passions outside work. Fantastic lecture. I learned a huge amount. Fantastic, and congratulations also on how much you contributed to not only to research but also the helping with the refined classification of these tumors. It's really amazing, and obviously to be involved in the WHO classification in itself is a fantastic achievement. So just encouraging people to put questions in the Q&A box, but I'll start with Damien Amendra's questions. He's got two. Excuse me. He says, if it's not too much burden, could you explain how methylation data can yield discrete copy number calls? And how do you set thresholds as to what constitutes copy number gain or loss?
Thanks very much for the question, Damien. Yes, it's a very useful technique that we get a 2-for-1 result from DNA methylation profiling, and it's because when we're doing the DNA methylation profiling, we're looking at over 850,000 different CPG sites across the genome, and so this can then be translated into, provided by all of those points being plotted into a profile, can give us copy number data associated with those particular chromosomes. And then via the different biological bioinformatical techniques, we can actually label and identify particular genes that we might be interested in, and then either look at the profile as a whole or focus in on particular genes of interest. And for my study, that was particularly interesting in looking at predicting potential fusions. And certainly we had the data to validate this via RNA sequencing and DNA sequencing of our fusion panel as well to look at these particular breakpoints. Certainly in terms of diagnosis, we tend to look more specifically at the copy number profile as a whole. And usually the threshold will be around a change of 4, the degree of 4 that we look at. But again, we also have NGS sequencing to help validate this as well. So usually it's a comparison between the two techniques to confirm particular losses of associated genes like CDKN2AB, which diagnostically is going to have a big impact on prognosis for some tumors.
Okay, and then a follow-up question. Do you have any clues about the role of the tumor microenvironment, which you may have come out through your research into the high-grade gliomas?
Yes, absolutely. So another really interesting question, Damien. Thank you. Yes, so one of the things I would certainly noted histologically, certainly with the TYA work, is the group known as the GBM mesenchymal atypical seem to have focal areas of lymphocytic infiltration associated with them as well. And so one of the interesting questions that's come from this is whether these immune cells have a particular role in this tumor. And one of the things that we can use, this abundance of methylation data that we have, is methyl cybersort. So I haven't run that through yet, but what that will tell us is the proportion of different immune cells that were in these tumors. And then using things like single cell sequencing on available frozen tissue, we can start to validate that and see what potential role that has within this as well. And I'm also doing some similar work on the infants with available frozen tissue with some collaborators looking at the single cell profiles of these tumors as well, and whether the immune microenvironment has any role to play in the future of these.
Okay, thank you. And a question from Mark Ahrens. What is known about how infant-type hemispheric glioma develops in the embryo/fetus? And is the fusion the key initiating and driving event, and what else happens during the progression?
Yeah, really interesting question, Mark, and something that's perplexed my brain over the years with these. Certainly, a significant portion of the cohort of these cases, these tumours were identified on the anomaly scan that parents would go to have to check everything was proceeding well with the pregnancy, and then unfortunately they would be told that their unborn child had a high-grade looking tumour. Certainly the fusion seems to be the driving alteration, because we weren't able, despite extensive sequencing of these cases, and that's been done clinically in the clinical setting as well, there were no other variants that were picked up. So, and certainly the data from other studies has suggested this is the important factor. As to what's actually initiating that and what's driving that, I am not sure, and particularly at this potential vulnerable age for them. And it also, to me, with the pattern of some of this high-grade tumor potentially differentiating and not progressing as would be predicted, it brings into question whether this is, in fact, could this be a developmental disease or some sort of problem that's happening in development rather than being a true cancer as well. So I think there's still a bit of an idea, or there's still a challenge in terms of understanding that. I think what would be really helpful is to actually have some of the tissue from that remaining actually static to actually confirm whether or not these are differentiating into ganglion cells, and also look at the expression potentially with the fusion. Has it potentially switched off at that stage, and therefore that's what's leading to the differentiation? But I think these are all hypotheses at this stage, but very interesting questions to consider if the research allowed in the future.
Thank you, Matt. I have a question. You've obviously had the opportunity to study these gliomas using different platforms. When it comes to the everyday diagnostics, are there recommendations that you could make for the test directory I mean, obviously there are always calls for improvement and revisions of platforms, and also targeting mutations or copy number variations. Do you feel that there is room for improvement for what is offered at the moment in the test directory?
Yeah, really important question, Sarah. And I think we all as a community need to look at this very carefully. Certainly, this study was one of many that contributed to now ensuring that the standard of care for pediatric brain tumors is DNA methylation profiling, DNA panel sequencing, and fusion panel sequencing as a standard. And I think before that, it was just an sort of ad hoc availability of that. So I think that's a significant progression forward. I think driving that forward in TYA patients is also going to be really important. Again, this study has highlighted the value of identifying things like hypermutators, but also things like the PDGFRA alterations. There are potential treatments that would be usable in that sort of context, as well as these potential mTOR inhibitors as well. So I think making these everybody who has a brain tumor and have availability of these tests should be something that we should be considering as well. But I also think thinking more broadly, thinking about things like the whole transcriptome, looking at the availability of whole genome sequencing. I think we're able to diagnose these tumors, but we're still at we're not advancing enough in terms of the treatment. And I think data of these different types being brought into the research community is what's going to really help with that. And I think pushing for those things, of course, it all comes down to finance and availability, but those are things that we should also be striving for in order to help answer these questions.
Okay, so a fourth question has come in, but unfortunately we've run out of time. Matt, if you do, and a fifth, if you wouldn't mind answering these questions, we can, we can record them. David Katz, we can obviously get back to, but with the anonymous attendee, we can't unless you provide us with an email address. But unfortunately, we do have to close things here. So thank you very much again, Matt. And just to let everybody know that there will be a ground round lecture in April. It will be given by Sian Morgan from Wales, who will be speaking about the genetic testing as part of clinical care in Wales. But the exact title is yet to be announced. So with that, I'd like to thank you all very much for attending, and please stay in touch and also show interest in perhaps attending one of our meetings, including the upcoming summer pathology meeting, which is a combined event with the BDIAP and the PathSoc. Thank you very much and have a nice day. Bye then.